over the opportunity to tell you about the biochemistry of fermentation and leavening.
Leavening is just when dough or batter or something like this expands because gas is being generated inside of it, and all those gas molecules are trying to escape, but they're getting trapped into the dough, and so the dough is just expanding. And this is how we can get things like bread instead of just flat crackers like matzah. Now, leavening can be caused by different things. So often we're talking about biological leavening agents, things like yeast. As we'll talk about a lot today, they go through this process called fermentation, which allows them to keep generating energy from sugar in the absence of oxygen. There can also be chemical leavening agents, so things like baking powder or baking soda, we'll talk a little bit about, as well as mechanical leavening agents, so things like those compressed air cylinders like Reddi-wip. But we're going to talk most about the fermentation today. And in order to understand the importance of fermentation, we kind of need to think about what it's achieving. The basics of fermentation is that it's going to allow us to keep breaking down sugar in the absence of oxygen. So there's a couple times this might come in handy. This might come in handy if you're in your muscles, say if you're working really hard, and it might come in handy for some sort of microorganisms and stuff if they're trying to grow when there's not much oxygen present. Now, in our muscle cells and stuff, if they're running low on oxygen, they're going to use lactate fermentation that we'll talk about. But in these microbes, they have another option. In yeast, they're commonly going to use ethanol fermentation. And this ethanol fermentation, it's going to give off gas, which is where we get our leavening, and it's also going to give off the ethanol. Now, when you're doing baking and stuff, you're going to bake off that ethanol, but when you're drinking wine and stuff that's not baked, that's where you get the alcohol from. But that yeast is not just making that carbon dioxide and making that ethanol just to get you drunk and full. Instead, those are kind of waste products. And the yeast's goal is really just catabolism, as much catabolism as possible. And so catabolism is molecule breaking, and it's one side of metabolism, which includes anabolism, so molecule making, and catabolism, which is molecule breaking. And a main reason why microbes or anything undergoes this catabolism is breaking down molecules to generate energy. Now, when we talk about energy generation and energy storage in biology and in biochemistry, we're typically talking about ATP and other what we call high energy phosphate bonds. So much more on this in other posts. But for now, just know that ATP is this molecule that can serve as a form of like energy money or energy storage. And so much more on why in other posts. But for now, just know it's kind of like an arcade token in that different organisms and cells and stuff, they can take in all these different food sources. So they can take in fat, they can take in protein, they can take in sugar, like blood sugar, glucose, and they can break it down and they can kind of like convert the energy in those food molecules into this storage form, this ATP. And then this ATP can be used by all sorts of different enzymes, so reaction helpers and all sorts of different things to power all sorts of different reactions. And so this ATP is going to allow your body and the body, or not the bodies, but the cells of those yeast and stuff, and to store energy for when they need it, and then they can use it for various things. So the goal when you break down molecules is, or one of the main goals is to generate ATP. Another goal is to kind of like generate intermediate products that you can use to make other things from. Now, one of the main molecules that gets broken down to form energy is glucose, so blood sugar. Now, even if you don't start with glucose, you can convert other molecules into glucose. And so glucose is often going to be the starting point for energy production. And what happens is you start by going through a process called glycolysis, where you break down glucose, and this gives you two molecules of pyruvate. So you're kind of breaking glucose in half, but you're not just breaking it in half, you're also modifying it a little. We'll go more into the terminology, but what you're actually going to be doing is you're going to be oxidizing it. When you oxidize something, you reduce something else. We'll go into what these terms mean. But for now, just know that this gives you, you're going to generate this molecule of NADH. You're going to take a molecule of NAD plus and generate a molecule of NADH. And you're also going to be, this NADH, you can't really see it here, but it's actually holding on to an electron. And this electron is going to be really important because the glycolysis, although you're generating 2 ATP, there's actually an important part of it is that you're generating this NADH, which has these electrons that can then go further down into this pathway, into this process called the electron transport chain. And those electrons can get passed off in order to generate even more ATP. And the pyruvate that was generated from glycolysis, it can go further into, it can get oxidized and go through this thing called the citric acid cycle. And this is going to generate even more of those NADH as well as FADH2. And all of those things are going to be these reduced things. So they're going to have these electrons that can then enter that electron transport chain and give you a lot of energy. So those electrons are going to be really important. That NADH is going to be really important. And all of this stuff is really important. But in order to go through this electron transport chain, we need oxygen. Because if you look, all those electrons, they're getting passed from one molecule to another. You can think of this NADH and these electrons as kind of like money IOU. You're getting this IOU that you have to then go take to another machine. And it's kind of like that, in that these electrons are going to be, and these electron carriers, so things like the NADH, like the FADH2, these are just going to be carrying those electrons and then passing them off, finally passing them off onto oxygen to generate that ATP in this pathway. Now, if you don't have oxygen, then you can't do this pathway. And if you can't do this pathway, well, then all of this stuff is going to back up and you're not going to be able to, say, regenerate that NADH and keep glycolysis going. And if you can't keep glycolysis going, you can't even get that wimpy payout of the 2 ATP from your glycolysis. And so if you don't have oxygen present, you need to have an alternative way of regenerating that NAD plus. And this is where refermentation comes into play. As I mentioned, there are a couple different forms of fermentation. And so there's fermentation that takes place through ethanol fermentation, and then there's lactate fermentation. We'll get more into ethanol fermentation in a minute. This is going to be where we're going to get our leavening, so our like bread rising and things like this, because what's happening is you're taking that pyruvate, so you're taking these carboxylate groups and making, getting rid of the carboxylate, the carbon dioxide, and then you have this acetaldehyde that then you're going to reduce to make ethanol. And again, we'll get more into what these various terms mean. But you can see that you're going from this kind of like double bond to an oxygen to a single bond to an oxygen. And this is going to make it so that you have this ethanol. Doing that requires these 2 enzymes. It's like pyruvate decarboxylase, which is going to get rid of the CO2. So decarboxylase gets rid of this carboxylate group. And then you have alcohol dehydrogenase. And so you can see that if you look from the ethanol's perspective, it's going this way, you're going to be losing that hydrogen, so it's dehydrogenating. I should mention that although I've drawn this arrows one way, all enzymatic reactions are, at least in theory, reversible. And so you can think about this reaction going this way, and this gives you how you can think of it as an alcohol dehydrogenase. But that's not going to be taking place in our bodies. Instead, in our bodies, we have lactate fermentation. And so lactate fermentation is going to play a key role in maintaining energy making when your muscles are working really hard and things like this, and they don't have time to have enough oxygen to go through glycolysis, to go through the citric acid cycle and go through oxidative phosphorylation and all this stuff that gets backed up in the absence of oxygen. And so instead, what happens is that instead of using those enzymes that those yeast have, we use an enzyme or reaction helper called lactate dehydrogenase. And what lactate dehydrogenase does is it's going to actually take that pyruvate and it's just going to give the electrons that were given to the NADH. It's going to give them back to this pyruvate in order to form lactate. So in the case of the ethanol fermentation, we were taking those electrons that we had put on the NADH, and we had placed them onto the acetaldehyde to make this ethanol. In the case of lactate fermentation, we're taking those electrons that we got from the NADH and we're placing them, that we put on the NADH, and we're placing them on the pyruvate directly in order to get lactate. Now, just a note, lactate and lactic acid, so you might see the terms used, and you can kind of use them interchangeably because these molecules are interchangeable. And which form it's in, the lactate version or the lactic acid version, is really just going to depend on the pH, which is a measure of how many of these protons are available. So these H+. If there's a lot of these H+ around, you're going to get this lactic acid form, which is this protonated form. Not a lot present, and you're going to get this base form, this lactate form. So it's just a pH dependent thing, and you're typically going to be having lactate. But you might see the term lactic acid used. Now, this lactate gives a pretty bad rap. So, but it's really not that bad. And so it's often blamed for like muscle soreness and things like that. Drives me crazy when I watch the Olympics because the commentators are all going on about this lactic acid buildup and stuff like this. But it's been shown scientifically that it's probably really not that lactate that's causing your soreness. It's going to be micro tears and stuff from all that hard work. And the lactate is actually playing an important role because it's allowing those muscles to keep going in the absence of sufficient oxygen. And it's not just a waste product. The liver can change that lactate back into pyruvate that can then enter oxidative phosphorylation to give you that big energy payout. So it's not going to be as efficient as if you were just to do it directly in your muscle cells, oxidate all that stuff in your muscle cells, because you have to go and transport it to your liver and all this stuff. But it's not like it's a total waste. And so you can still get energy out of those pyruvate molecules. So don't blame the lactic acid for your soreness, and I'll post some links to more information on that. Let's now go into a little more detail because I promised to discuss a little more about these like oxidation and reduction and all this stuff. So what we need to know is that oxidation is going to be the loss of electrons and reduction is the gain of electrons. And I have whole posts on this whole like redox thing. So check those out if you are interested in more. But basically atoms, so all those oxygens and hydrogens and carbons and all those things, they're made up of even smaller things. So they're made up of subatomic particles. We have protons, which are positively charged, neutrons, which are neutral, and electrons, which are negatively charged. And the protons and the neutrons are going to hang out in this dense central nucleus, and then around them is going to be whizzing all these electrons. Molecules can do things like share electrons to form bonds, these covalent bonds, or they can give and take electrons. And when molecules give up electrons, we say that they get oxidized. And when they gain electrons, we say that they get reduced. And now we could either be talking about like gaining or losing whole electrons, or more commonly in biochemistry, what we're talking about is kind of just like bonding to something that's going to be hogging the electrons. In the case of oxidation, you're binding something that hogs the electrons like an oxygen. In the case of reduction, you're kind of binding something that you can steal more of its electrons from, more of the shared electrons from. And so something like a hydrogen is going to be easier to pull electrons away from than an oxygen. And so if you have a bond to an oxygen, it's typically an oxidation reaction. And if you have a bond to a hydrogen, that's typically a reduction reaction. If we look at NAD plus and NADH, remember how I said that a bond to a hydrogen is kind of like getting electrons because you can pull the electrons away from the hydrogen. And so it makes sense that this NADH is going to be this reduced form. And what you have is, so you have with NAD plus, this is the oxidized form. With NADH, you're going to have the reduced form, and you're going to have this NADH kind of be holding on holding on to an electron that you can then pass off later in the chain. And so when we're doing these fermentation processes, what we need to be doing is we need to be taking that reduced form and we need to be oxidizing it. And in order to oxidize the NADH, we have to reduce something else because oxidation and reduction are flip sides of the same coin. So in the case of the ethanol fermentation, we're going to be reducing acetaldehyde. And in the case of the lactate fermentation, we're going to be reducing pyruvate. And both of these things will regenerate that NAD+, and this NAD+ can then be used to help power more of the glycolysis. But you get different products. So with lactate fermentation, you're going to get lactate, which remember can then be converted back into pyruvate in your liver to make energy from through the more traditional oxphos pathway and stuff like this. And in the yeast and stuff, you can get this ethanol fermentation that is going to make it's going to give off gas, which is going to be our leavening, which we'll talk about, and it's going to give off alcohol, so it's going to be this ethanol. Let's talk a little bit more about leavening now. So remember that leavening is just when your dough or your batter or something like that is going to be expanding due to gas generation. Gas is basically just a state of matter, and so anything can be like a solid, a liquid, or a gas, and some molecules are more likely to be in one form than another at a certain temperature. Basically, if the molecules really like one another, they're going to be really sticky to one another, and it's going to take more energy for them to break apart. So they're going to need to get hotter in order to form a gas. But some molecules, things like carbon dioxide, like that carbon dioxide that we're generating in our yeast fermentation, those molecules don't really like each other that much. And so they're happy to get away from one another. And when they get away from one another, then we say that they're in this gas form, when they're kind of whizzing around with one another. And they're kind of whizzing around and they want to get as far apart from one another as possible. And so you're going to have things expand. The more gas molecules you have, the more expansion you're going to get. This is something called Avogadro's law. And so your volume is going to be increasing as you're generating more of that gas. And this is going to be happening inside of bread dough or whatever. And so the gas molecules, they can't just fly away. Instead, they're kind of trapped within this network of proteins within the bread dough. Those proteins are called glutenin and gliadin, which together make up what we call gluten. So gluten is really just these proteins, these bread proteins. And they form this kind of like meshy network. And there's going to be this gas being generated inside of this meshy network. And this meshy gooey network is going to be like this flexible. So it's going to be able to expand, but the air can't get out. So that's why you get those kind of like air pockets and stuff. And when you heat it up, basically all those proteins are kind of going to gelate and solidify, and they're going to hold in that expanded structure. So all those holes and stuff, that you get this nice fluffy bread. And so that's going to be what's happening with these biological leavening agents in the case of fermentation. So you're generating the gas based on this fermentation. And then with other types of leavening agents, you're going to be generating it different ways. And just a couple notes. So when we're talking about the ethanol or fermentation, so like yeast and stuff like this, they're going to be breaking down glucose, as we talked about, but they don't get the glucose directly, because what's going to happen is that the plants store glucose not just as individual glucose molecules, but they store it as these starch molecules, which are kind of these chains of glucose molecules. So you can think of it kind of like, instead of just having a bunch of pennies all over the place, you have like rolls of pennies or something like this, and you have to break those open, break them apart in order to spend them. And so the sugar starch has to be broken down, and this can be done by these enzymes called amylases. Now, flour actually has these amylases in them because the flour's grain kernels need to be able to break down that starch when the plant wants to use it. And so they have this amylase that can then break down that starch into smaller pieces. And then the yeast breaks down those smaller pieces into individual glucose units using maltase and sucrase enzymes. And then they ferment that to make the gas from. And remember, this fermentation process is going to be regenerating that NAD plus that can then be used to keep glycolysis going and allow for further energy production. There are other ways to generate gas, though. And one of these is with chemical leavening agents. So here we're talking about things like baking powder and baking soda. So what's the difference? Baking soda is just the base, and baking powder is the base plus the acid plus cornstarch to keep them apart until you're ready. So an acid is something that donates a proton, so one of those H pluses we were talking about, and a base is something that steals a proton. And so you can get an acid-base reaction, but with sodium bicarbonate and carbonic acid, that is going to then generate this carbonic acid that can break down into water and carbon dioxide. And so you have this process where an acid and a base are reacting, generating carbonic acid, which can then generate carbon dioxide. With baking soda, you just have that base, and so you're going to add an acid, something like buttermilk, brown sugar, yogurt, lemon juice, vinegar, cream of tartar, molasses, applesauce, or honey. And in the case of the baking powder, it's going to have a base and an acid in there. But it's also going to have cornstarch or something like this, basically to keep them apart so that the base and the acid aren't reacting before you want them to. Because we don't want them to react and give up and kind of be wasted before we actually have them ready to use. In order to kind of make it so that they're only used when we want them, you typically see dual acting baking powder. So they actually have two different acids in there. One of them is something like monocalcium phosphate or MCP, which is going to be fast acting. So it's going to give up those protons quickly. And so it's going to rapidly react with the baking soda when you get them wet. So you basically just add water. Remember, the cornstarch is going to kind of be keeping things apart. But when you add water, you can get these to rapidly react. It's going to release about 60 to 70% of the available CO2 within 2 minutes. And it's going to be kind of generating these mini pockets in the bread. So not like big bubbles, like those big holes, but kind of smaller ones throughout. This is going to set the stage for the slow acting acid, which is going to require heat. And so when you heat this up, after you get then sodium, the aluminum sulfate, this like slower acting agent that is then going to give you those more of that leavening, more of the rising. And so again, this is just taking place through this acid base reaction where you have this base. When you add an acid, that base gets converted into carbonic acid, and then that carbonic acid is going to break down to give you water and carbon dioxide, so that's CO2 gas. And this is the same CO2 gas that you get when you break down the glucose, when you break down the pyruvate through this process of ferment. And this is the same CO2 that you would get if you were to break this down through ethanol fermentation. But in the case of the biological leavening agents, you're also going to make this ethanol, but remember that gets burned off when you go and break that bread. Hope that helped you understand fermentation and leavening and all this fun stuff. And happy cooking if you're cooking, and happy Passover if you're watching this on Passover, and happy all that stuff.